available. Emma, can you go ahead and swap in?
Yeah, sure.
Awesome. Thank you so much. So she's coming from Institute de Neurosciences in Barcelona in Spain. And she will be speaking on cannabis-based therapeutics, a novel multi-target approach for Leigh syndrome. And I'll turn it over to you whenever you're ready.
Okay. Hi, everybody. First of all, I'd like to thank the Mito Foundation for inviting me to give a talk. I'm very honored to be part of this wonderful symposium. And today I'll talk about cannabis-based compounds for the treatment of Leigh syndrome. Over the last 20 years, I've studied the effects of cannabinoids in the brain. Cannabinoids are compounds that are exclusively present in the cannabis sativa plant. And when I saw the pathology of mitochondrial diseases, including Leigh syndrome and some of their hallmarks, it became clear to me that one of these cannabinoids, cannabidiol, I'm sure you heard about it, could be of interest for Leigh syndrome. And why? Well, when mitochondria fail, there is oxidative stress and free radicals are generated, and CBD is antioxidant. There is also neuroinflammation, and CBD is anti-inflammatory. And there is neurodegeneration, and CBD has been found to be neuroprotective. Moreover, in contrast to other cannabinoids such as THC, CBD is not psychoactive, which is very important in the context of mitochondrial diseases, which mainly affect the pediatric population. And last, there is a large body of evidence suggesting that CBD has a higher safety profile. And you might know that CBD has been already approved for the treatment of the epilepsy associated to 3 other rare diseases: Dravet syndrome, Lennox-Gastaut, and tuberous sclerosis. So for all these reasons, we decided to study the potential beneficial effects of CBD in this syndrome. And the results we found are very remarkable. So I'll start showing you the results we obtained using the mouse models and later using patient-derived cells. So we first used one of the gold standards mouse models, the NDFS4 knockout mice that Simon nicely introduced. So these mice have been well validated by many labs. They closely recapitulate the human disease. And for example, they have a very short lifespan, a reduced body weight, failure to thrive, many motor and respiratory alterations, seizures, and also neuroinflammation in specific brain areas. So we started treating these mice with CBD daily, and we found that CBD treatment extends their lifespan and improves associated comorbidities. So here on the left, I'm showing you the survival graph, and in gray, you can see that these knockout mice have a very short lifespan. They die around postnatal day 55, and when they are daily treated with CBD, they live much longer. Moreover, we found that the CBD treatment delays the appearance of some clinical signs that have been associated with neurodegeneration, such as curling behavior or clasping behavior, as I illustrate here in this image. So we did a more complete characterization of CBD's effects in this mouse model that you can find in our recent publication in Nature Communications. So as I mentioned earlier, CBD has been approved for the treatment of the epilepsy associated to 3 other rare diseases. However, no one has studied yet whether CBD can also exert anti-seizure effects in Leigh syndrome preclinical models. So given that epilepsy is a common manifestation in Leigh syndrome, that some patients are treatment resistant, and that some classical anti-epileptic drugs, such as sodium valproate, are not recommended due to their mitochondrial toxic effects, we decided to study the potential anti-epileptic effects of CBD in mitochondrial epilepsy. And to do so, we took advantage of another mouse line. In this case, it's a conditional knockout mouse line of the same gene I told you before, NDFS4, but in this case, instead of being deleted in all cells, it's been deleted only in a specific neuronal population, the GABAergic cells. So these mice have a fatal epileptic seizure, and they usually die when they are around 2 months old due to these fatal attacks. And as you can see here in this graph, when we treat these mice daily with CBD, we significantly extend their lifespan. Actually, we double their lifespan. So one hypothesis is that CBD is increasing their lifespan because it's delaying the onset of these fatal epileptic attacks. So we decided to study a little bit more into detail these anti-seizure effects. And instead of waiting for these spontaneous fatal attacks to occur, we just induced the seizures. And we can do that by thermally inducing them. So we can just apply a heat map on these conditional knockout mice and trigger the seizure. That's a bit similar to what happens in some Leigh syndrome patients. When they have a fever, they're more likely to have seizures. So the first thing we observed was that CBD increased survival after seizures. Why is that? Does CBD protect against the negative effects of seizures, or does CBD reduce the seizures themselves? Well, we found that CBD indeed reduces the number of tonic-clonic seizures. And in fact, acute CBD treatment can reduce the duration and the intensity of seizures that are already ongoing. So in addition to epilepsy, we also used the same conditional knockout mice to study additional hallmarks of the disease. For example, we addressed the social deficits because there are some patients that have autism. And we can study that in our mouse model by using the 3-chamber social interaction test, where we just place our mouse into a maze and we let this mouse to freely explore another mouse that has never seen before or an object as a control. So mice are social creatures, and we can just record the exploration time and then calculate the discrimination index. The higher it is, the better social skills mice have. So as you can see here, our conditional knockout mice have lower discrimination indexes, around 0, meaning that they have social deficits. And remarkably, those conditional knockout mice that were daily treated with CBD had similar social skills as healthy mice, suggesting that CBD can rescue the social deficits associated to this mouse model. So far, I've told you about CBD's effects on behavioral symptoms, but we've also studied its effects on cellular and molecular hallmarks of the disease. And as an example, I'd like to show you what we found regarding neuroinflammation. So there is a nucleus named external globus pallidus that is part of the basal ganglia. And this is highly affected in both patients and our mouse model. And as you can see here in these immunofluorescence images, on the left, we use a marker for astrocytes, and on the right, a marker for microglia cells. So in both situations, there is an increased reactivity in the external globus pallidus of our conditional knockout mice. And interestingly, this is totally normalized with the daily CBD treatment, suggesting that CBD decreases neuroinflammation in this highly affected brain area. So I've shown you some of the effects we found in our mouse models, and I also wanted to share some of the results we gathered using Leigh syndrome patient-derived cells. So we used the fibroblasts coming from skin biopsies of 2 Leigh syndrome patients that have mutations in 2 different genes. So the first thing we did was to confirm that despite having different mutations, these 2 patients have similar molecular profiles. So we did RNA sequencing studies, and we found several genes that were commonly downregulated in these 2 Leigh syndrome patient cells compared to healthy fibroblasts. And interestingly, these regulated genes were normalized by the CBD treatment. And the majority of these genes were related with antioxidant processes. So it seems that CBD, on the one hand, can exert antioxidant effects due to its chemical structure per se, but additionally, it can upregulate certain genes that are involved in the antioxidant defense. But are these molecular changes translated into a reduction in oxidative stress? Indeed, they are. we used a probe called Mito that labels a reactive oxygen species level. And by using this probe in the mutant fibroblasts, we found, first of all, as expected, that the cells from these two patients had higher levels of ROS levels. And second, that CBD treatment reduced the ROS levels in all conditions. This reduction in ROS levels was correlated with an amelioration of the phenotype of these cells, because the mutant cells proliferate slower, which makes sense, right? Their mitochondria fail, they have less energy, so they just grow slower. And when we treated them with CBD, we found a slight increase in their proliferation rates. So overall, it seems that CBD also improves the phenotype of these Leigh syndrome patient-derived cells. So I've told you what CBD does in several preclinical models, but we've studied also what it does, how it does this. And I don't have a lot of time to go into the details of the CBD's molecular mechanisms of action, but here is a list of some of the key factors and signaling pathways that we found that were dysregulated in Leigh syndrome and that CBD could somehow normalize. And I like to highlight PPAR gamma because we found that this receptor was downregulated in both mouse models and patient-derived cells. And interestingly, we also found that CBD can act as an agonist of this receptor and promote part of its therapeutic effects. So I believe that these broad effects that we found are largely because CBD has a polypharmacology effect, and it can act at multiple levels at the same time, in contrast to other existing drugs that basically target one factor, one signaling pathway. So just to sum up what I've told you so far is that CBD improves the survival and fitness of Leigh syndrome mouse models and ameliorates the phenotype of patient-derived cells. These effects involve multiple mechanisms, including PPAR gamma activation and the attenuation of neuroinflammatory responses and oxidative processes. So our study provides preclinical evidence suggesting that CBD might represent an effective treatment for Leigh syndrome. However, caution must be taken, and I've just shown some results regarding preclinical models. We are now in the process of founding a university spin-off that will be called Therapeutics to fundraise and be able to sponsor a clinical trial with CBD and Leigh syndrome patients. In Madrid, there is now a small study under off-label compassionate use of a few Leigh syndrome patients that are currently taking CBD, but the idea is to be able to sponsor a bigger trial. But in addition to that, we are highly motivated in developing novel treatments with a greater efficacy than CBD itself. And for that, we're just combining CBD with other molecules that exert synergistic effects with CBD. So we started doing high-throughput screening in yeast mutant models, the ones that Ethan just showed. Actually, we do this with Perlara, and we combined CBD with the Pharmacon library. And by doing so in these yeast mutant models, we found 13 rescue hits. And now we're going to just validate these hits in C. elegans models of Leigh syndrome, brain organoids, and in the mouse models I just showed you. But in addition to developing better drugs, we're also highly motivated at identifying novel biomarkers for Leigh syndrome, which we believe might be very helpful for clinical trials. So we are just combining our omics data coming from proteomics, transcriptomics, from several models, and using artificial intelligence, we expect to find common signatures. And then we'll validate these biomarkers using Leigh syndrome patient samples from 2 key hospitals in Spain. And last but not least, we're also using mathematical models combined with informatics in order to perform some clustering, correlating the symptoms of patients with their specific genetic mutation for patient stratification. So we're highly motivated working in these 3 lines of research, and we're very excited about the upcoming results. And finally, I just would like to thank all my lab members, who have greatly contributed to the results I've shown you, and also the funding agencies and the patient associations, especially Cure Mito Foundation. And finally, my lab is part of a big group called Mitochondrial Neuropathology Group, where Albert Quintana is one of the group leaders. He's my former mentor and now a close collaborator. And I also would like to thank Marcello Belushi, who is the neuropediatrician in Madrid in charge of these off-label compassionate use studies with the Fievoli syndrome children and CBD, and also Alessandro Prigione and Ethan Perlstein for their contribution in the CBD-based combination drugs that we're working on. And thank you all for your attention.